Pleasure to introduce Stephen Mondo, who works, fungal and algo team at JGI. So Stephen received his PhD from State University of New York at Bingham. All right. Okay. Thank you. Thank you. And his PhD from Cornell University. He joined JGI in 2014 to have fun with fungal genomics. And area of special interest to him is epigenomics and chromatin organization. And today he'll talk about periodic DNA patterns associated with chromatin regulation across eukaryotes.
Okay, hi everybody. And I'd like to start by just thanking the organizers for providing me this great opportunity to share some of my newest work with the community. So as mentioned, I'll be talking about these interesting DNA patterns that we found associated with nucleosome organization in eukaryotes. So a little background on nucleosomes. These are the basic unit of DNA packaging in eukaryotes, and they're incredibly conserved. So across the billions of years of evolution of eukaryota, these nucleosomes have remained virtually unchanged. And what they are is a complex of two different main components. You have a histone protein complex made up of 4 different types of histone proteins, each duplicated once to make a symmetric octamer. And then the other component to the system is DNA. About 150 base pairs of DNA wraps around these histones to form one nucleosome. And then these nucleosomes are joined together to form larger structures through a variable amount of open chromatin or linker DNA. And so as you can imagine, with their primary purpose being to package information away and store it in small spaces, the positions of these nucleosomes are going to have major consequences for things like gene regulation. You could think about a transcription factor trying to access its binding site. And if that binding site is hidden away inside a nucleosome complex, it's going to be very hard to express that gene. And so in that context, it becomes really important for us, if we're to understand how eukaryotes regulate gene expression, we need to understand how these nucleosomes are being regulated, what kinds of dynamic processes exist to shift nucleosome positions, and are there any kind of static regulators that kind of fix nucleosomes in one position in the genome. And so, here comes the slide. So there's been a lot of work done on these dynamic nucleosome regulators. These are things like chromatin remodeling complexes and epigenetic machinery. And I put this figure up here from a recent review on histone methylation, just to illustrate kind of the depths of knowledge we have on some of these types of regulators. So all of these little gray circles are different modifications of histones that people have described. They know generally how they are associated with gene expression. Are they activators or suppressors, et cetera? However, there is another component to the system that I think comparatively has received much less attention, and that is the DNA itself. And so the idea here is that as histones are DNA binding proteins like transcription factors, is it possible that they have preferences for the types of DNA that they interact with? Are certain sequences or shapes more preferable for binding than others? And so there is some precedent for this. So back over a decade ago, most of this work was done. And for example, in Saccharomyces cerevisiae, they describe what they call intrinsic DNA code for nucleosome placement, where they claim that the vast majority of nucleosomes in vivo are positioned by these high frequency shifts in dinucleotides. Other groups have characterized these kinds of favorable DNA patterns that are flanked ideally by repelling elements that help to kind of statically lock a nucleosome in one position across many conditions. And so while this is really exciting for potentially understanding gene regulation and for potentially genetic engineering, when people try to take these observations and move them to other model eukaryotes, there didn't seem to be all too much agreement, or these patterns were not really present. And so this led to a general lack of enthusiasm to try and explore this area deeper. However, now fast forward to today, over a decade later, we're well within the genomics era. We don't have to really limit ourselves just to these model eukaryotes quite so much. And so is it possible that we stopped too early, and there are more conserved, important histone DNA relationships that are just waiting to be discovered? And so this is where I got interested in the project. I started by looking at start and end coordinates of a variety of different genomic features for any sort of DNA patterns. And so the one I'll be talking about mostly today is the start of transcription, the 5' UTR start. And what we settled on was looking at per site GC frequency surrounding all of these features. And I'll show you what that looks like in just a moment. And we did this for all published and freely available data through the Mycocosm fungal portal and Phycocosm portal that Igor mentioned earlier. And so here's just an example of one of these plots down at the bottom. So again, this is Saccharomyces cerevisiae. The x-axis is position surrounding the transcriptional start site in this case, and the y-axis is %GC. So this is a summary across all genes in the genome. And so the colors represent upstream and downstream of the feature, the TSS, and the black line in the middle is a smooth curve showing kind of the overall denoised profile in that region. So you can see there's not all too much going on here. It's a relatively flat distribution, aside from a little dip in GC upstream of the TSS. But here's what you see in some of the more understudied lineages in eukaryotes. And this example is Leucosporidiella creatinivora. It's a distant relative to rust fungi, very poorly understood clade. And you see this very different pattern where you have this consistent shift in GC that's occurring surrounding the transcriptional start site. And the distance between these peaks is roughly nucleosome sized at about 150 base pairs. And so we wanted to kind of understand the breadth of this pattern, both within genomes and phylogenomically. And so we developed a very simple method to just calculate the strength of this signal, which we're calling a periodic DNA pattern score, where essentially we just took the heights of all the peaks that are observed surrounding these features and added them together. And so in the case of Leucosporidiella, the score is about 0.7, whereas in Saccharomyces, it's 0 because we don't see this signal. And then you learn a couple important things when you place this into a phylogenomic context. First is that it seems to be enriched, particularly at the transcriptional start site relative to other genomic features. And so the size of these circles here are proportional to the score that we calculated. And the colors represent different features, with the TSS being in this dark green. The other thing we see is that these patterns are non-randomly distributed across eukaryota. So we have an enrichment in the green algae, the chlorophyta, as well as in the basidiomycete fungi, particularly the microbacteriomycetes, where this Leucosporidiella lineage is from. And so because these are nucleosome-sized patterns at features we know it's important to have proper nucleosome phasing, we wanted to look further at this question to see if they actually are related. And so we worked with our sequencing group who developed an approach called NucSeq, where they take genomic DNA, just naked genomic DNA, and add histones to the mix and allow the two to naturally self-assemble in vitro before they digest it with micrococcal nuclease. And we sequence the nucleosome-bound fragment of DNA. And so this is a great way to get a physical map of the preferences that histones have for certain DNA patterns, because there's not really any epigenetics in the system beyond DNA modifications potentially, which I'll talk about in just a moment. So we did this for 5 fungi and 1 alga. And what we found is that these patterns are actually highly correlated with nucleosome positions in vitro. And so again, I'm showing you positions surrounding the transcriptional start site. And on the y-axis, I'm showing you in green the AT frequency. And in orange is this in vitro occupancy that was calculated using this NucSeq approach. And I've scaled these curves by standard deviation from the mean, so you could see them on the same scale. And what you see is that the two are really tightly correlated, especially in the lineages with these high periodic DNA patterns, and those scores are in the parentheses, particularly the basidiomycetes, but we tend to lose this profile in lineages without these patterns. But in Chlamydomonas reinhardtii, green algae, where we have kind of a moderate score and we know epigenetic modifications are present kind of in this region, we still see these peaks in AT kind of having some relationship to in vitro nucleosome organization. And so, of course, in vitro is not real life. It's entirely possible that in vivo epigenetics rules and these signals are totally wiped away. And so we wanted to explore that question as well. And so we took advantage of a few different types of data. We used in vivo MNA-seq data sets, which is essentially NucSeq except done in vivo. And then we also took advantage of ATAC-seq data, which is kind of the opposite. It sequences the open chromatin in the genome, so non-nucleosome bound fragments. And so what we see is that, in fact, in vivo, these nucleosome or these AT patterns have a strong influence on nucleosome positions. And so here I'm showing you the same plot that was on the previous slide, except now I've overlaid a map of the nucleosome-free spaces surrounding the TSS. And you can see the regions where we have these high amounts of AT and nucleosomes are bound in vitro. We have very little open chromatin, telling us that there are nucleosomes present there. And this pattern was consistent across different data types and different lineages. In general, telling us the profile that the more AT you have, the more likely there will be nucleosomes at that position. And so we wanted to take advantage of this to develop new methods to potentially predict nucleosome occupancy based just on DNA alone, because these kinds of experiments are somewhat challenging. And so I don't have time to talk about how we developed our training set, but we put together a deep learning model that combines a convolutional and recurrent neural network to predict these in vitro occupancy profiles. Per nucleotide, we're predicting occupancy in the assembly. And so here's just one example of an organism we excluded from training so we can look at the overlap between actual in vitro nucleosome positions and our predicted nucleosome occupancy profile. And you can see the two are really nicely correlated. So we're doing a pretty good job of recapitulating what's happening in these fungi. We also found that these predictions can have meaningful impacts in vivo. So here we took advantage of an RB-tDNA-seq library, which is essentially, it's a TN-seq approach, more or less, where you have a random insertion mutagenesis library across your whole genome. And we looked at this. This is from Rhodosporidium torreoides. And we know that foreign elements, CRISPR-Cas9, transposons, et cetera, struggle to integrate into nucleosome-bound regions. And when we look at this tDNA-seq library, which is in magenta, you see that it's unevenly distributed across the genome. But this predicted nucleosome profile that we calculated can kind of explain that uneven distribution because they tend to be avoiding nucleosome-bound regions. And so with that, I'm going to quickly wrap up and say that we found these periodic nucleosome-sized DNA patterns in eukaryotes. They are enriched in specific clades, and they are enriched at the transcriptional start site. They are correlated with nucleosome positions, both in vitro and in vivo. And we can build methods based off of these to predict nucleosomes more accurately in eukaryotic genomes. And so I'm hoping to use this as a starting point for developing more advanced rules that are governing histone DNA relationships. The idea is that now that we have a kind of profile of where histones should prefer to bind, we can look in vivo and see where the discrepancies are and try and learn what epigenetic regulators are there to explain that difference. And so with that, I have a lot of people to acknowledge, but I particularly wanted to highlight Guifeng, Igor, Ronan, and Hugh from the JGI, and Kathy Aim from Purdue University. And I'd be happy to take any questions if I'm not over time.
Thanks a lot for an interesting talk. I was wondering if you could take the same approach to screen archaeal and bacterial genomes, like many archaea are known to encode histones, if you see the same patterns.
Yeah, that's a great question. And I haven't looked at archaeal or bacterial. Archaeal will be very interesting to look at because I know they have something like akin to nucleus or histones and nucleosome complexes.
Yeah.
And yeah, that would be really cool to explore, but I haven't gone there quite yet. We're still kind of at the broad level.
But in principle, the same.
In principle, yeah, absolutely. We could take a look at that.
Thank you.